from molecular to the genomic scale. So yeah, let's get started.
Thanks for the introduction, and sorry I couldn't join in person. There's a lot of my colleagues there who I'd love to see in person again, but maybe sometime soon. My name is Brian. Thank you for having me speak, and I'm excited to share about the work that we've been doing on training genomic foundation models. But as we'll hopefully see, these models, even though they're only trained on genome sequences alone, they can get some sense of multimodality and other interesting aspects of biology from raw sequence. And I wanted to start with this quote, actually, by Frances Arnold, where she says, "Today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it." And so if you think about it, there's been tremendous advances in technology for genome sequencing, genome synthesis and assembly, and even genome editing. But it's still very difficult for us to control biology. We still suffer from many complex diseases. It's hard for us to engineer complex biological systems. And fundamentally, this is because it's hard to compose or write in the native language of biology. And in part, this is because this code is incredibly complex. So if you think about it, all of life uses DNA to encode the complexity of biology that we see, and this complexity is written in a very simple code of A, C's, G's, and T's, but it's completely unintuitive to us as humans. The human genome is 3 billion base pairs long. It kind of boggles the mind. I probably look at DNA sequences almost on a daily basis, and I probably still couldn't tell you the difference between a random string of A, C's, G's, and T's and the genome sequences that come from an actual organism. But there is information in these sequences, right? And how do we actually extract this information and learn higher order rules from it? So we've been inspired by models that have made tremendous progress in learning higher order rules from, say, human natural language. And fundamentally, these models are trained with very simple objectives, like predicting the next token in a sequence of tokens, where a token could be an English word, or a token could be a DNA base pair. And a very important idea is that even though next token prediction seems like a very simple task to train a machine learning model, actually, if you train the model on a sufficiently large and complicated data set, then potentially next token prediction or next word prediction in language, for example, can encode many very complicated tasks within it. So for example, a good next word predictor in language would not only have to at least simulate some understanding of grammar or semantics, but also facts about the world, or how words are associated with human sentiment, or translation between languages, or spatial reasoning, or math. Potentially all of these concepts are encoded in next word prediction in language. And even though these large language models are not learning an explicit world model, the world has left its imprint on text from the internet, for example. And so a model that's trained on text from the internet can at least simulate some understanding about the world. So what about in biology? Well, in biology, we typically like to think of these two general domains. We have genotype, which is a DNA sequence, and this sequence encodes for complex phenotypes via some biological mechanism. And it's often these mechanisms and these phenotypes that we want to understand in biology. But unfortunately, in many cases, it's very hard to collect high throughput data on biological phenotypes or mechanisms. And machine learning models, especially modern ones, rely on very large, rich data sets. In contrast, it's very easy to collect data at the level of genotypes. And this is due to advances in things like next generation sequencing. So what if we could train a model on this breadth of genotype data and get some information about mechanism or phenotype? And fundamentally, this could potentially work because even though evolution acts on phenotypes, the results of this evolution get written back into genotypes via natural selection. So in the same way that the world has left its imprint on text from the internet, the world has also left its imprint on DNA sequences through evolution. And so a hypothesis could be that a very simple task like next base pair prediction of a DNA sequence could actually teach a model many complex tasks about biology. For example, which mutations are correlated with health or with disease, or how these nucleotides form codons and how these codons come together to encode proteins, or how bases pair with each other to form RNA structures, and maybe even how genes come together to make biological systems. So this would be a very attractive hypothesis if it were true, right? You could train a model with a very simple task and teach it a breadth of different things about biology. But is this actually true in practice? A first step that many in the AI for biology community have taken is not training language models on sequences of DNA, but training them on sequences of proteins. And these are protein language models that are trained to predict the next amino acid in a sequence, or a related task where you fill in the blanks and predict masked amino acids given unmasked amino acids in the sequence context. These models are, again, trained on hundreds of millions of proteins across basically a snapshot of all observed protein evolution. And the idea is, evolution has rules, and maybe some of these rules are written into sequence variation of proteins from which you can learn higher order information. And I won't talk that much about protein language models here, but suffice to say that these models that are trained on large databases of protein sequences and structures have revolutionized the study of molecules. So, for example, methods for protein structure prediction, like AlphaFold, methods for de novo protein design, won the Nobel Prize in Chemistry in 2024. And this is a very well-deserved prize. Molecules are very complicated, and we're still very interested in molecules. But we're also interested in how these molecules come together to make a complete organism, for example. And even a very simple single-celled organism is orders of magnitude more complicated than any individual molecule. So how can we now teach machine learning models things at this level of complexity without losing molecular resolution? Well, luckily, over the past 3 decades or so, we have actually been sequencing the complete genomes of millions of organisms and depositing these DNA sequences into publicly available databases. And in theory, embedded in all of this data is information that's required to create whole organisms. So can we actually extract this information using machine learning? So this brings us to a model that we put out last year. It's a model that we're calling EVO, or EVO1 for the purposes of this talk, and it's a DNA language model. And fundamentally, all this model is doing, it's doing a very, very simple task. All this model is doing is you give it a sequence of DNA base pairs, and it predicts the likelihood of the next base pair in the sequence. That's all the model is doing. The model itself, though, is very large and very complicated. So it's a neural network with 7 billion parameters. The model is also trained on 300 billion tokens, where a token is a single DNA base pair. For EVO1, we restricted training to simpler genomes of prokaryotes as an initial proof of concept. And there's 2 important things that we want to extract, or 2 kind of important hypotheses we wanted to test with EVO1. So the first idea is that even though we're only training a model on DNA sequences, because of basically the beauty of the central dogma and how evolution or biology, encodes information with the same fundamental code, that DNA sequence directly codes for RNA, and that RNA directly codes for protein. Especially in prokaryotic genomes, the protein coding sequences are mostly contiguous. So one idea is that even if we're only training a model on DNA, we potentially get RNA and protein modeling for free out of the model. And another important idea in the EVO1 model is that even though we're only training a model on DNA sequences, if we increase the length of the sequence that we can have the model process, then not only will we be able to have the model understand information about a single gene, but also how this gene interacts with the broader multi-gene context, and in some cases a complete genome. EVO1 could process an input sequence of around 100,000 base pairs. And we trained a 7 billion parameter model because we observed what's also been observed in other domains like language and vision. It's this idea that if you increase the amount of compute used to train the model, then the model's ability to predict the next token also kind of magically and predictably increases, or it gets better. So you can see on the x-axis of this plot the floating point operations used to train the model. As you increase the amount of compute, the model's ability to predict the next token in terms of perplexity gets better, so lower complexity is better. So scale is one dimension you can use to improve language modeling performance. The other is architecture. So we actually use an architecture called Stride-Taina that outperforms the traditional neural network architecture that's typically used for language. So once you have a DNA language model, what can you actually do with it? So one class of tasks that we like to think about are roughly what we call prediction tasks or interpretability tasks. And so here we're doing a genetic perturbation experiment, but instead of perturbing the genetic sequence of an organism, we're going to perturb the inputs to the neural network and measure the effects on the output likelihoods from the neural network. And the perturbation we're going to make in this case is we're going to insert a premature stop codon into all of the coding sequences in a prokaryotic genome. So premature stop codon insertion is typically very bad. It would make sure that this, if in the organism, it would make sure this gene does not code for protein and effectively knock out the gene. And interestingly, what we saw when we did this experiment on EVO1 is that when we put a premature stop codon insertion into an essential gene, EVO's likelihoods on average go down by a lot. But when we put a premature stop codon insertion into a non-essential gene, EVO's likelihoods don't change all that much. So the model actually has learned an understanding of which genes in a genome are more tolerant of these very bad mutations and maybe which genes are more or less essential to the organism. This is without us giving the model any explicit information about which genes are more or less essential. We didn't even give the model any information about where in the sequence there was a gene, that the model had to learn all this information from raw sequence. Another thing this class of models can do is generate new sequences in the same way that ChatGPT can produce new code or write an email for you. EVO can generate new DNA sequences. So in this case, for example, we took the model, we specialized it to generate CRISPR-Cas systems that are used for genome editing. These are very complicated systems. Cas9 is over 1,300 amino acids. It's also bound to a guide RNA. And actually, so EVO could produce this sequence we called EVO Cas9-1. And when we chemically synthesized this DNA and tested it, the molecules that this DNA sequence encoded had comparable in vitro cleavage to SP-Cas9, which is the state-of-the-art system for genome editing. But importantly, the model had less than 80% sequence identity to anything in nature, or the Cas9 that was generated had less than 80% sequence identity. For context, this is enough to break any patents around CRISPR-Cas, and so these molecules are novel, at least in the eyes of the US Patent Office. We then wanted to push the novelty of this model even further, and for this we actually ran a very interesting experiment. So here, we leveraged the fact that the model can learn information across multiple genes. So you can prompt the model with DNA that encodes a gene and get DNA in the output that also often encodes a gene. And in prokaryotic genomes especially, genes with functionally that are functionally related are often next to each other on the genome sequence. So we can basically prompt the model to generate regions of the genome of interest by giving it some of the native genomic context. And we ran an interesting experiment, which was what happens if you prompt the model to generate a region of the genome that's often very hypervariable, or that often has a lot of evolutionary novelty. So for example, we prompted the model to generate a region of the genome that often encodes these genes called anti-CRISPRs. These encode for, say, a protein that inhibits CRISPR, for example, by binding to Cas9, for example. And so we prompt the model with, say, an anti-CRISPR associated protein, for example, and generate genes in its output. And we find when we test these genes in the lab that many of them also have anti-CRISPR activity. But then we can compare these Evo-generated anti-CRISPRs to all the genes that we've ever discovered from nature. And a couple of these designs actually have no significant sequence similarity to anything in nature. And the sequence composition of these genes are more consistent with de novo designed proteins, say from the Baker lab, than trivial recombinations of natural proteins, and they retain anti-CRISPR function. So we see this as substantial evidence that these models are not just memorizing. They can actually non-trivially generalize out of their training distribution and actually come up with some pretty creative sequences that have functions of interest. We then prompt the model to generate 100 billion base pairs of AI-generated genomic sequence spanning thousands of putative functions and make these sequences publicly available. The last generative task we did with Evo1 is instead of generating a single gene, we then generated 1 million base pairs. And we thought that these genome scale generations would not be reasonable at all, but actually along some metrics, they look somewhat reasonable. So for example, the predicted coding density is what we would expect. We find tRNAs that cover all 20 canonical amino acids. So we compare this to maybe the first versions of image generation models where they would generate blurry pictures. Maybe the first iteration of these Evo models can generate blurry genomes, but there's definitely room for improvement. So then we were interested in expanding this paradigm to eukaryotic genomes. So this is a very important domain of life. It includes plants and animals, but also humans, and so we want these models to also be useful for human genetics tasks. The problem is that eukaryotic genomes are in the billions of base pairs instead of the millions of base pairs. They also have very complex genetic architecture, lots of non-coding sequence, so we needed to do a lot of work first in data processing. So a few months ago, we put out this model called Evo2. It's a model trained on all domains of life, spanning bacteria, including insects and plants and primates, including humans. We did a lot of work in data processing, especially for the eukaryotic genomes, to make sure we were training on functionally interesting parts of the genome. We also did a lot of work in scaling these models even further, so Evo2 can now process an input sequence of 1 million base pairs. To do this, we actually had to go back to the architecture and make additional improvements to enable the model to utilize GPUs at 1 million base pairs somewhat efficiently, at least. So this is an architecture we're calling Stractina 2. It's especially hard to scale models to 1 million base pairs when the model itself is also very large. So Evo2 was scaled to 40 billion parameters. We had to do a lot of engineering for this. Greg Brockman was on a sabbatical from OpenAI and helped us train these models. And NVIDIA graciously donated 2,000 H100 GPUs to help us train this model. The total amount of flops going into training Evo2 makes it actually the largest model ever trained on biological sequence data by compute flops. We also make this model what's called fully open. So we open source not only the model weights, but also the code for training the model, the code for running the model, and the training data. And actually, by this standard, we think EVO2 is the largest fully open model ever released, including across modalities like language and coding, for example. So once we have this model, what can we do with it? Well, again, we can do these perturbation experiments where we mutate the input to EVO2 and measure the effects on the model's likelihood. But now we can do this on, say, human gene sequences. So in this kind of benchmark, we apply EVO2 to human variant effect prediction. So all humans have some genetic variation, and most of the time this variation is completely benign. But in rare cases, it does lead to disease, so predicting those variants is very important. We hypothesize that if you have high likelihood under EVO2, then maybe those variants are correlated with health, or at least with compatibility with life. But if you have very low likelihood under EVO2, then maybe those variants are correlated with disease or loss of function or incompatibility with life. And very interestingly, on this data set called ClinVar, which are variants that have clinically relevant effects, it's compiled by the NIH, we did not actually develop EVO2 as a variant effect predictor for humans. It's just trained on these species spanning all domains of life. But actually, the model is quite competitive at variant effect prediction, even compared to models that are specifically designed for variant effect prediction. And actually, in the subset of variants that are not these single letter substitutions, so these are variants like insertions, deletions, duplications, these non-SNVs, EVO2 actually outperforms all the models that we tested on ClinVar. The model can also do this for these variants of BRCA1, which is a gene that's often implicated in breast cancer. The model is, again, competitive with the best models for coding single nucleotide variants, and in some cases outperforms all models that we test in non-coding single nucleotide variants. Beyond interpreting the model as a black box, we can also peer into its representations. This is inspired by some work done by a company called Anthropic on a model called Claude. And they actually find a part of the representations of Claude that only activate when it receives inputs related to the Golden Gate Bridge. So we do the same kind of interpretability study on EVO2 by training these sparse representations. And we find dimensions in these sparse representations that do activate only on, say, regions of the genome that code for non-coding RNAs, or protein level features like alpha helices and beta sheets, or TF motif features as well. Anthropic found a feature that would only fire on bugs in your Python code. We actually find a feature that fires on frame shift mutations. So this kind of interpretability can find errors in computer code and genetic code, for example. And beyond this, EVO2 is also a generative model. So it's not even meant for scoring or gene annotation. It's actually meant as a generative model. So we wanted to make this model useful for much more complicated design tasks than, say, single gene or single protein design. So for example, we wanted to see if these models could generate complete genomes that were viable. So we first applied this to the design of bacteriophage genomes. So these genomes are small enough to be tractable, but they're still very complicated. If you're doing whole genome design, you not only have to design all of the genes, there's also non-coding, regulatory elements. The genes and these non-coding regions all interact with each other. The genes also interact with genes that are not on the genome. So suffice to say that whole genome design is very complicated. We also picked phages because they're an emerging therapeutic modality for multi-drug resistant bacterial infections. This is a famous case from a few years ago. Tom Patterson almost died from multi-drug resistant infection, but his life was saved by a phage, and actually he made a full recovery, and this reignited interest in phage therapy in the United States. The particular phage we wanted to target as our first kind of design template is this phage called 5x174. It's actually it's very famous. It's the first DNA genome that was ever sequenced by Fred Sanger in the 70s. It was the first genome chemically synthesized by Craig Venter and team in 2003. And so we wanted to make the kind of the first AI-generated genomes inspired by this historic genome. It's also very complicated and very compact. It has 11 genes, but some of the genes actually overlap in the DNA sequence. So they share the DNA sequence, they're just out of frame with each other. So it's a very compressed, very difficult genome to design. So to design variants of 5x174, we first specialized EVO1 and EVO2 on microviridae sequences. So this is the family of phages that are around 5x174. We can then prompt the model with the first few bases in the 5x174 genome to generate variations of these genomes that we can then, again, chemically synthesize and test experimentally in the lab. And this experimental testing yielded 16 functional phages. Actually, one of these phages has less than 95% average nucleotide identity to any known phage. So if we had discovered a phage like this in nature, it would technically qualify as a new species. There's a lot of very interesting things about these phages that I don't have time for, but one interesting thing that we saw is that there's one phage with interesting structural changes. So we solved a 2.9 angstrom resolution structure via cryo-EM of this phage called EVO536. And it actually used a much smaller J protein in its capsid. And interestingly, another lab had previously tried to integrate this exact gene. So this is a J gene from a different evolutionarily distant phage. They had tried to integrate this J gene in 5x174 previously and had failed. But actually, EVO was able to integrate the gene and make compensatory mutations in the genetic background that enabled this gene to actually work and produce viable phage. We then pool these phages into a cocktail and show that the cocktail can also more rapidly overcome bacterial resistance than a single kind of monoclonal, phage. This is important because there's a rise in bacterial infections that cause diseases that affect humans and a lot of agricultural diseases, and there's a lot of phages that could potentially be useful therapeutics in these cases, and we're actually interested in seeing some of our AI generated phages could actually make a difference in the clinic. Cool. So that concludes the data I wanted to present. Hopefully I've convinced you that even though these models are being trained really with a very simple task, all these models are doing is predicting the next base pair in a sequence. Actually this very simple task can generalize to prediction and design tasks that span genes to genomes. So for example, we can predict how single nucleotide changes affect the function of this breast cancer gene. We can segment different regions of the genome and annotate them using representations learned by the model. We can predict how individual genes are more or less essential to the entire organism. We can also design these de novo anti-CRISPR proteins. We can design these multi-gene systems like these CRISPR-Cas systems involving protein and RNA. And we can design complete genomes at the scale of complete bacteriophages. And I wanted to, again, return to this quote by Frances Arnold, where at the end she does say that evolution has learned how to compose DNA sequences. And so the way we think of this research is that there's already technologies for reading, writing, and editing DNA. And now, as we get better at composing new DNA sequences, potentially at the scale from genes to genomes, then this will finally enable us to control the complexity of biology much better and enable, say, genome design. So all of this work is documented in publications or preprints. The references to those are on this slide. We also make all of our models and code for running the models, training the models, everything we do is out in the open, totally open source for anyone to use freely, and we're big believers in open science. Finally, this is work done by a tremendously diverse and talented team spanning many different areas of expertise, many different institutions. EVO 2 was produced by ARC Institute, Stanford, NVIDIA, a collaboration with the labs of Patrick Hsu at ARC in Berkeley, Hani Goodarzi at UCSF, some collaborators at the University of Washington, a startup called Goodfire for doing interp. And finally, my lab also led the work you've seen here, and I'm very grateful to be working with such a talented group of students and postdocs. And so that concludes the talk. I don't know if there's time for questions, but if there is, then happy to take them.
So we do have time for a couple of questions.
Hi, Brian. Absolutely beautiful work. My name is Bin. I developed a phage language model called MegaDNA. And my question is about the phage genome generation part. I saw you used both EVO1 and EVO2. For EVO1, you used a long context version instead of the short context version, which is 8K. So are there any rationale behind these choices? And what do you think is the best model to generate this kind of new phage genome? Thank you so much.
Yeah, it's a great question, and I'm a big fan of your work as well. So it's good to see you. So we started with EVO1 because when we started this project, EVO2 had not been developed yet. But kind of that's the reason why EVO1 is in this project. But then we saw that the model fine-tuned on EVO2. That model is also probably a better model, just as a base model. So we actually just use this isn't the preprint, but we do a head-to-head comparison of the base models between EVO1 and EVO2, and the EVO2 base model actually does much better at phage generation, like without any fine-tuning, than any of the EVO1 models we've ever trained. So I would definitely recommend EVO2 as a more powerful base model. The fine-tuning can get you, though, like if you are able to do fine-tuning, you can even like rescue the performance of EVO1. But for most cases, I would definitely recommend EVO2. That's just going to be a better model.
Thank you so much.
Hi, everyone. I'm Zais from Princeton, and I have one question about the controllable generation of the DNA based on the EVO series models. So I think to generate some functional proteins, the controllable generation is quite important. And I see in your paper you may use some beam search method to guide the generation. Have you tried other methods for more precise controllable generation? Thank you.
Yeah, that's a great question. I think that's like the big open question. So the thing, yes. So we have a very capable generative model, but naively, right, this model will just generate unconditionally, and maybe that's less useful. Ideally, you want to do conditional generation towards the function of interest that you want. So we've been doing, so for example, we can show that if you prompt with genomic context, that's one way of specifying or conditioning the model, is you condition it with like the genomic context of interest that encodes your function, and that's one way of steering the model towards towards functions of interest. The other way that you alluded to, which is in the EVO2 preprint that I didn't have time to present, is coupling the model with another model that can also guide it towards functions of interest via inference time search, inference time steering. That's another great way. But all the methods that we've kind of at least published on don't involve any additional training of the EVO model itself. But obviously, if you want to do even better controllable generation, then we probably also need to explicitly train the model to do conditional generation better. And from this, you can probably there's been a lot of work on this in text, for example. So ways of conditioning on specific prompts, ways of fine-tuning the model, RL-inspired fine-tuning. I think this will be a very interesting and is a very interesting open area for these models. It's a great question. Yeah.
Okay, thanks for your answer and the presentation. Thank you.
Hi, Brian. I'm Yuan from Beijing Zhongguancun Academy. Thank you very much for your presentation. So my question is related to the previous one. I'm very curious about how you designed the prompt for DNA generation, especially in the autophage example, and if there is any experiences you would like to share when designing the prompts in DNA generation.
Yeah. So a lot of this is in it's a great question. The prompting strategy for the model actually requires some amount of engineering, like prompt engineering. So we showed that the model can respond to genomic context, and based on the genomic context, that can actually have a big role to play on whether the model just regurgitates its training data or whether it is able to make these creative variations on the training data. For the phage prompting, it's interesting that you bring this up. So we fine-tune the model and we still prompt with the first part of the 5x174 genome. We found if you prompted the model with not enough of that context, the model would just generate random phages and nothing that was close to 5x174. But if you prompt with too much of the 5x174 genome, then the model would just regurgitate the literal 5x174 genome and not produce any variation on it. So there's this sweet spot, and it was somewhat weirdly different for EVO1 and EVO2, where the model there's enough conditioning to generate something that looks like phi-x, but the prompt was also short enough to encourage the model to produce novelty in its generation. So we basically just do a sweep of different prompt lengths and have these in silico functions to score. But it's an interesting finding that prompt engineering, which is very important in large language models of text, is also very important in large language models of DNA.
Thank you very much.
Maybe a quick final question, only because we don't have, meet the speaker
okay.
session for.
Yeah, hi, Brian. My name is Jie Cong. I'm from Changping Laboratory. And also, I'm a big fan of this EVO too. So I got a quick question. It's I would like to hear your comments about the autoregressive way to change such a kind of EVO. So we kind of using this autoregressive inspired from ChatGPT, but sometimes the genomic language kind of have a different grammar, right? For example, in human language, every word might have its meaning in the sequence, sentence. But in the DNA sequence, we have lots of redundancy. For example, some TF binding site, they might have several redundancy across the genome. But using the next token, you kind of treated every base have the same weights during the loss function. So I really like to hear your comments. Is there any we need to redesign this kind of loss function, or maybe have another diagram to change this kind of large language model in the genome? Yeah, thanks.
Yes, I'll keep my response brief as well. But yeah, the playing with the loss and architecture sometimes to deal with the loss is going to be very important. Yeah, autoregressive models, they train really well. They're very stable. It's a very rich information, a way of providing the models with information. But it's true, some parts of the genome sequence, especially in proteins, maybe has a left to right order. But especially as you get to eukaryotic genomes, maybe you need some bidirectional context. Maybe you need to train with a discrete diffusion loss instead of next token. Maybe you do need to. So actually, EVO2 does do some loss reweighting. So non-coding or more repetitive sequence gets weighted less than functionally rich sequence. So we do play with that a little bit in EVO2. But in general, yeah, potentially though, next, autoregressive modeling has taken us really far in DNA, surprisingly far, but it is definitely worth, especially for some tasks like structure prediction or long-range interactions, thinking about losses that are more like that encourage bidirectional context.
Okay, thank you.
Thank you. Unfortunately, I don't think we have time for more questions. I'm sorry.
It's a very short question.
Sorry?
It's a very short question.
Okay, go ahead, but we have to keep the schedule moving.
Yeah, sorry. I think he's out.
Maybe you can email the question to
Thank you,
him.
thank you.
Let's thank Brian again for taking
Brian.
time to join us, and I think just email him. Yeah, let's get moving to the next
Okay,
talk.
thank you.
Thank you, Brian, for joining us remotely and for taking all the questions. Let's welcome our next speaker, Dr. Henrik Nielsen, who is an associate professor at Technical University of Denmark, and he'll be talking about signal peptide prediction.